Thank you very much for the invitation. I'm currently not the CEO of Health in Code, but most of the work I will show you has relation to this, to my previous participation in this company. So let's talk about the clinical interpretation of next generation sequencing data. These are my potential conflicts of interest. Next generation sequencing really has changed our ability to explore the genetic causes for diseases, because we can now evaluate big number of genes, whole exome, and even the whole genome with our parallel sequencing. And we have gone from this situation, Sanger sequencing, to the possibility of study the genome with apparatus or machines like the HiSeq. 65,000 sequencers, Sanger sequencers, are needed to do the same job as now we have with one next generation sequencing machine. So we go from the classical approach, fix the gene, to the current approach, fix everything. And in this situation, we will find unexpected but relevant findings, the lobsters, but we will find, of course, and we will catch a lot of variants, genetic variants that are not relevant and maybe even toxic. So the main challenge now in next generation sequencing is the interpretation, with thousands of variants in each individual requiring classification and interpretation. For the interpretation, the first is to establish, of course, the pathogenicity. And for this, we use basic information, molecular information, and type of mutation. Mutation is truncating the protein, it's just a change in one nucleotide. Does it change the amino acid encoded in the original protein? We evaluate the conservation of the affected nucleotides or genetic regions in the evolution. We can do other bioinformatic studies, and there are different predictors based in the structure of the proteins and other parameters. We can do functional studies expressing the mutations, for example, in cellular models, and sometimes we have animal models that are relevant for confirmation of pathogenicity of genes or defining the potential mechanism of the conditions. But most important usually is the clinical information. And the clinical information, the most important or most useful for filtering variants is the presence of the variant or its absence in healthy individuals. And happily now we have a lot of big cohorts of control individuals that have been sequenced, and then we can exclude the things that are common in the population. We have to evaluate always co-segregation within the family, but we have to consider different inheritance patterns, and we have to consider the previous descriptions of the genetic variants or similar genetic variants that may have been described in other families. So when you have a genetic variant, the first thing is whether the variant is pathogenic or non-pathogenic or something intermediate, likely benign, likely pathogenic. So this is difficult, and most interpretations finish here in this categorical classification. But this is not sufficient. Of course, if the variant is non-pathogenic, then we have to consider whether we need to study more genes or to evaluate more variants or to finish the study. But if the variant is pathogenic, the first question is whether the variant explains or not the phenotype. And for this, we need previous information about what is the phenotype related with the variants. If the variant is not only the phenotype of the patient, we have to know whether the variant explains the phenotype of the family. If it doesn't explain that, then we are in the first situation, consider other genes or end of the study. But if the variant explains or supposedly explains the phenotype of the patient and the family, we have not finished. Now we need to look for the available information on the variant. And here I will concentrate my talk. The clinical value of genetic testing is completely dependent on this last step. So what is the clinical information we have to look for? What is the age of diagnosis of the disease? What is the penetrance? What are the morphological and functional consequences that we can expect from the variant? And what are the clinical manifestations? What could be the complications in this patient? Are there any therapies or preventive measures we can take? And this means that we need prognostic information. And the only way to get prognostic information is to recover the information that is available and to do additional studies and research. So knowledge management is essential for this step. We have data that are dispersed about diseases, patients, families, genes, mutations that are in many, many different papers, Congress meetings, presentations like this. And we need to transform more of this information or this data in information and knowledge that we are going to apply to an individual patient. So for that, our strategy for many years has been that, okay, we are going to read all the papers and try to attend all the conferences. And we have people introducing, experts that are introducing the data, the relevant data in databases. And from these databases, we will be able to try to approach a clinically oriented report. So this, as I told you, now I'm not currently working in Health Code, but this work has been done in this company that has been oriented to do this work. And at the time I did this slide, we have evaluated more than 30,000 papers with more than 62,000 families. And now I know there are more than 200,000 individuals that have been collected. So it's not only to collect the individual and the mutation, that there is a patient with the mutation. We have to collect all the family information that is available and all the clinical data that are relevant for each phenotype. So how we work with this? With the database, we can query the database. And here, for example, we are querying missense variants in beta-myosin. We have non-synonymous variants in this gene. And here we were able at this time to collect likely or very likely variants, rare variants, I mean mutations here, in more than 3,000 patients from 1,600 families. So we can do survival curve from birth because we consider all the individuals that are in the pedigree. And here you have the variants and then the survival curves. And what you see here, almost 3,000 individuals with data on follow-up available, including patients and relatives that are clinically affected or are carriers of the genetic variant, are even not clinically affected. And here you see, for example, at age 20, you have 95% survival. So we can compare and we can start evaluating what is the profile of different genes. This is in orange, myosin binding protein C. There is a difference, myosin binding is better, but the difference is not huge, but it's highly significant. And here what you see is the light blue, this is the percentage of death causes. Blue light is sudden death. Young individuals usually die suddenly. Older individuals, you have in pink, heart failure death, stroke-related death. Similar for both genes. Other genes, this is troponin T, 1,000 individuals, worse. Troponin I, 500 individuals, worse. Tropomyosin in purple. Myosin light chains 2 is better, lower number of individuals. Myosin light chain 3 in pink is in the worst, one of the worst. Actin, cardiac actin gene is here, better survival. And you see the difference are, all of them are significant difference. Troponin C, maple green. GLA, Fabry disease, is the best for hypertrophic cardiomyopathy. This is only cardiovascular death or transplant. And this is PRKAG2 in brown. This is hypertrophic cardiomyopathy and Wolff Parkinson White cardiac glycogenosis. And this is LAMP2 Danon disease, the worst. This is hypertrophic cardiomyopathy in young individuals and progressing to dilated cardiomyopathy or dilated cardiomyopathy in females. So you see different genes, different prognosis. So there is information, prognostic information here. It's not only the gene. Here we compare males and females. This is males with Danon. When you are 30, 40% survival. When you are 40, 15% survival. These in brown are females with Danon disease. It's much better than males, but it's worse than any of the other. And these are usually dying of heart failure. 40% survival age 50. This in red is Fabry in females, and this green is Fabry in males, significant difference. And these are X-linked genes, so it's normal that males are worse than females because they only have the abnormal copy. But this is beta-myosin heavy chain in orange, females, and in blue, males. And you see, if we consider from birth, the prognosis is worse and significantly worse in males. We live less than the females. So the prognosis is not, of course, dependent only on the genetic variant, on the gene. It's depending on the genetic variant, because if you consider that in any gene there are variants that are non-pathogenic, you cannot say the prognosis of the gene. And this is very important when you read the guidelines and they say, if you have this gene, you have to consider a defibrillator. So this is beta-myosin, the whole gene. And this is one particular mutation in beta-myosin, glycine 716R gene. And you see the prognosis is completely different. At age 20, with this mutation, you have a 20% sudden death incidence. And at age 50, you have 15% survival, and these are heart failure deaths. So this information, only 42 individuals. So sometimes we do not have enough number of individuals to evaluate the prognosis of an individual mutation. But we can then try to compare the prognosis of mutations that are similar or are in the similar genetic region. So this is what we did here. These are the protein regions affected by glycine 716R gene. So it is in the gene, in the whole protein. It is in the head of the protein, in the myosin motor domain. It is in one region that is the converter domain, defined by different ways in different databases. And this is a domain that is important in fourth generation in the beta-myosin. And it is in one particular helix defined in these protein databases within the converter domain. So we can evaluate those different regions. And you see here, this is the whole gene, all the missense mutations. These in red are missense mutations in the converter domain. 711, 756 are the amino acids encompassed by this domain. These in green are the mutations in the helix, 715, 721, and this is our mutation. So what you see here is clearly within the converter domain, there is a region that is very relevant. And it's interesting because this region is target of the new drugs for treatment of hypertrophic cardiomyopathy. Other example I will show you with troponin T is not only the beta-myosin, are any gene. All the genes have the same behavior. So if you take troponin T missense mutations, we have a lot of patients, and here with 674 patients, we were able to collect, this is the survival curve. Age 50, 70% survival. In young individuals, sudden death, older individuals, more heart failure death, and other events, other cardiovascular causes. This is a family that was described by McKenna and collaborators in 1990 with the presence of mild or absent hypertrophy, but a lot of sudden death, premature sudden death in very young individuals. The ECG was abnormal, but the echo was quite normal, except if you consider the atrial dilatation. And there was a lot of disarray. So in this family, the genetic diagnosis found the mutation, the arginine 94, which is in a hotspot domain. And if you take this hotspot domain, 92 to 95, that we have identified as a high-risk region, you see age 50, 40%, almost 40% incidence of death, most of the sudden death, and when they are 60, 45% survival. But all the troponin T is the same. We have, for a long time, we have read that troponin T is a gene with a high incidence of sudden death with mild hypertrophy. Not all the mutations are the same. This is a mutation, asparagine 271 isoleucine, that we have a founder effect in Galicia and Portugal. And you see, with 140 individuals from different families, the survival is very, very good. No death until you are in the 30s, and a much lower incidence of death than the troponin T hotspot. So not all the troponins are the same, not all the troponin T. And if we compare males and females, here you see these are females with the benign mutation 271, males with the same mutation, actually quite worse. These are females 92, 95, and these are males 92, 95. A huge difference is not only the gene, it's the genetic variant, and of course the gender is important. In dilated cardiomyopathy, I will show you another example of how this works. The prognosis depends on the genetic cause, of course. These are titin truncating variants. More than 1,000 individuals collected, most of them from the literature. And here you see sudden death is interesting because in individuals from 10 to almost 70, sudden death is 50% of the deaths. So these people may die suddenly. Other question is they don't die suddenly when they don't have manifestation, clinical manifestation of the disease. So these are truncating pathogenic variants, usually variants in the A band or in highly expressed exons. So here what you see, the difference, if you put males and females, they are different. Females are better. Here we compare with beta-myosin heavy chain mutations that are related to DCM in orange. So titin is better if you have dilated cardiomyopathy than to have a beta-myosin heavy chain mutation of the cause of the disease. This is lamin in green. So important, lamin may be a high risk gene. It is indeed. If you are in the 60s, the probability you are alive is approximately 40%, putting all the pathogenic variants. But here, if you compare, so when you are very young, lamin, they don't have a high incidence. So until you are in the 30s, the probability of death is approximately 5%, similar to titin, cardiovascular death. This is filamin C in red. So intermediate between titin and lamin. This is desmoplakin, maybe arrhythmogenic, but also hypertrophic. This is emerin, it's worse. This is LAMP2 that also causes dilated cardiomyopathy. And here you have BAC3 in gray. So different prognosis with different genes. RBM20 is also one of the worst. Now, if we compare males and females, this is a bit complex slide. It's different. So here, for example, you see this is titin. We have seen the difference that females are slightly better than males. But for example, lamin is males and females, unexpectedly is very similar. And here in red, if you see filamin C is much better in females than in males. Males with filamin C are very similar to lamin. So the ESC guidelines for heart failure, August this year, they talk only this slide about the genes and the consequences of the genes. And what they say is lamin, RBM20, phospholambanam, FLN, I don't know what is this gene. They say they have higher risk of sudden cardiac death. Early indication for primary prevention by ICD implantation should be considered. And they say guided by risk factors as detailed. They don't detail the risk factors in the guideline. We have to go to the 2050 to see some risk factors. For example, here in lamin mutation, the risk factors, they say non-sustained ventricular tachycardia, ejection fraction, low ejection fraction, male gender, non-missense variants, and they say that if you have two of these, you are at risk. For example, male and non-missense. Let's see lamin mutations again. Here you have the survival of the whole population, more than 1,000 patients. And here you see this is a missense mutation, arginine 190 tryptophan. It is even worse. And this is a missense mutation. Not all the missense mutations are like this. There are others that are better. Of course, most of the truncating are the same. Yeah. So we have males and females. They are similar. And if you see truncating filamin C mutation causing this arrhythmogenic phenotype encoded by filamin C with this MRI, and there's a lot of arrhythmias and sudden death, if you compare males and females, are completely different. Females, young females, have very low risk, and males have a high risk. Sudden death, all of them. This is the age of onset. Females, when they are 50, less than 30% have clinical manifestations. But you see here, if you are 50, there is a diagnosis of 35% of males and 52% of females. And there is sudden death in 10% of females and 35% of males.
We are running out of time. Sorry to interrupt.
So, filamin C, put the gene correctly, pathogenic variants, only pathogenic, truncating variants, early indication. What is early indication? And guided by risk factors. Be careful. The missing point is the variant. My take-home message, the interpretation does not finish in a categorical classification, benign or pathogenic. The prognosis depends on the genes, and genetic variance and knowledge management is necessary. Thank you very much.
Thank you.
Thank you very much for this excellent talk and the databases that give us prognostic evaluation. A quick question from the audience. Did you reclassify the MUHA7 variants according to the updated MUHA specific guidelines by ACMG?
Sorry, you are talking about the ESC heart failure guidelines?
Yeah, the guidelines. There was one guideline now for one gene. As an example, MUHA7, beta myosin heavy chain, and for missense variants, for instance, and the location of the missense variants. I think this is what Felix Boschan means.
I guess what the question is about whether we should reclassify and explain in the guidelines about this. And the problem is it's very complex. I don't think in the guideline that try to summarize in the 100 pages, we need more pages. So we need a different approach for the interpretation of genetic testing clinical setting. And we cannot say early defibrillators should be considered in lamin because there are variants that are doubtfully pathogenic, other variants are really bad. But in filamin, it's even worse. Females without clinical expression, they don't die. Suddenly, you don't have to put a defibrillator. But a male, if you don't do, so we need this information to go to further detail. But the problem is most people is not thinking when they publish about mutations that they have to give the clinical details and that you have to study the family. And without that data, we cannot establish the prognosis.
Exactly. So maybe just one quick answer. How do you communicate this information that you have now from these databases to the clinicians and to the patients in writing? Like who
in this case,
the approach communication work.
No, in this case, the approach for us it was to create a company to be able to dedicate people to do this job and then provide the information in the reports. It will be nice to have it for free, but someone has to pay for that, and this has never been feasible for. And so the only approach for us was to. But the problem of the publications is that all we are using here, most of it is already published. The problem is who is compiling the data and putting together and doing the analysis.
Exactly, and we have to work on this together.
Yeah.